Hi and good morning, everyone. Today I'm going to take you through my research on metagenomics. I want to start out by breaking down the title of this presentation into its core concepts. So the aim of this part of my research was to use metagenomics to highlight food safety risks in irrigation water sources. To put it broadly, metagenomics is the study of genetic material recovered directly from environmental samples. In my research, I chose next generation or Illumina sequencing. By food safety risks, I'm speaking about pathogenic or toxic organisms with negative human health consequences. Did I just get booted?
You're all good, Emily. Just, yep.
You've asked me to share my screen, I'm sorry.
Yep, no, it's all good. You're right.
Okay. And to get even more specific, the water sources I'm talking about is water and sediment from the Upper Hawkesbury River. So for next generation sequencing, this produced millions of strings of bacterial DNA from my samples. These DNA sequences were investigated using two approaches. The first one was to align the sequences to a database of known organism identities with the aim of identifying as many species as possible, including human pathogens. The benefit of this approach is that we can circumvent the need for performing numerous individual detection assays for each species we are interested in. And the second approach was to predict the organism functions. We can infer from our sequences what the functional capabilities are, meaning the actions and interactions of the bacteria, regardless of their assigned taxonomy. These predictions can capture a diverse range of functions, including their role in human disease pathways. So the first of our discoveries was that metagenomics successfully revealed human pathogens and other food safety risks in our samples. Highly abundant in our water sources were key groups of bacteria relevant to human health. These include proteobacteria, Bacteroidota, and cyanobacteria. Narrowing down further to which specific genre or species we could detect with this metagenomics approach, we uncovered a range of potential risks. You can see on the screen now a list of taxa of great concern to the fresh produce industry. On the left are some well-known pathogens, and on the right are some cyanobacteria, also known as blue-green algae, that can potentially produce harmful toxins. This is an emerging area of fresh produce safety research. Of course, detection in the water using metagenomics does not always mean detection on or in the crop, but this is a concern nevertheless. Moving on to the functional predictions, a majority of the bacteria in our samples play a part in metabolism and information and cellular processing. However, this approach also predicted that our samples contain genetic material known to play a role in human diseases, such as antimicrobial drug resistance. The presence of these human disease pathways predicted by this approach does not necessarily mean that the bacteria in the water are capable of causing these human diseases. It could be a case that we are picking up on human contamination in the water, and this is a remnant of that. So I hope I've been able to convince you that metagenomics is a useful addition to traditional methods of pathogen identification. Metagenomics is able to present a snapshot of the microbial risks found in irrigation water. As Chana mentioned earlier in her speech, treating water is a challenge for the industry, and this is especially true here in Australia, where our water sources are limited. For this reason, irrigators should understand these potential hazards in order to reduce the burden on post-harvest food safety measures. And finally, I really want to stress the point that natural water sources are under the influence of all users, whether they're industry, agriculture, or communities, and therefore should be viewed holistically. This is also known as the One Health approach, and it highlights the link between public health and the fresh produce industry. I'd also like to acknowledge the traditional custodians upon whose ancestral lands this research was conducted and from which the samples were taken, the Darug people, and pay our respects to the elders past, present and future.